Hi everyone, I am Gangothri and today I am going to present my work titled The Changing Diet for Male House Mice Over the Generations Influences the Female Mate Choice. Now, sexual selection is one of the driving forces that ensures the reproductive fitness of an organism. Apart from the genetic factors that can influence the reproductive fitness, there are also various environmental factors that influences the sexually selected traits. Now, when an organism experiences such shift in the environmental conditions, they show plastic adjustments to these shift. And when these adjustments are being reflected on the sexually selected traits and they are being passed on to the next generation, this forms the basis of sexual selection. Now, from various environmental factors, one of the prime environmental factors that influence the sexually selected traits is resource availability. That is the diet that the organism has been provided with. Now, there have been studies which have shown the how shifting diet can influence the reproductive fitness of an organism within the generations, but not many studies have seen the effect between the generation, which is exactly what we ask in our study. Does the changing food quality alter the sexually selected traits in males intergenerationally? And for this, we used mice as the model organism and we studied the behavior of female mate choice towards the different male mice to study the characteristic of how diet can influence the reproductive fitness. As for the diet, we provided different populations of mice with high quality food treatment and standard quality food treatment from F1 to the F3 generation and switched the diet for half of these populations in the middle of the F3 generation and tried to see how that would reflect on the reproductive fitness in the F4 generation of these male mice. Now this is how the female mate choice setup looks like, where the males from F4 generation with different food quality treatment were kept in the satellite cages and the female was kept in the central cage, which was allowed to move and visit all the male cages. We ran this experimental setup for 24 hours and we had total 24 setups like these. And with this, we try to see by the end of the experiment if the female is making a decision for a male from particular food treatment or not. This is how the actual experimental setup looks like. Now, when we did the analysis and compared the amount of time that the female is spending with a male from different food quality treatment at different time points of the experiment, this is something which we got. During the first hour of the experiment, we did not see a preference by the female towards any male, which is what we were expecting. During the second half of the experiment, that is 12 to 24 hours, we saw that the female is significantly spending more amount of time with the males from pure high quality treatment as compared to any other males. And to see if the female is making a choice during the last hour of the experiment, the third graph shows that the females are significantly spending more amount of time with the males from pure high quality treatment as compared to the other treatments. That is the standard quality or the males that have been switched from high to standard quality or from standard to high quality. Now with this study, we try to address two hypotheses. First is the silver spoon hypothesis, which states that when an organism experience ample amount of resource availability, that would lead to an increase in the fitness of an organism. And the second hypothesis, that is the match-mismatch hypothesis, addresses the intergenerational aspect that we are trying to study in our experiment. This hypothesis states that when an organism experiences a shift in the environmental condition, that would lead to a reduction in the fitness between the generations. And with our female mate choice experiment, we saw that the males that did not experience any shift in the food quality, that is the pure high quality treatment, were significantly preferred more by the females as compared to any other food quality treatment males. And to address the match-mismatch hypothesis, we showed that the males that experienced a shift in the food quality treatment, be it from high to standard food quality or from standard quality to high quality, led to a decrease in the reproductive fitness of these males in the F4 generation as compared to the pure high quality males.
Thank you.